Hi everyone, it's Wednesday, it's lunchtime, so it must be time for another University of Sydney's Lunchbox Science. Welcome everyone to our webinar today. I'd like to invite you to wash your hands first of all and crack open your lunchbox. And for those of you who are joining us for the first time, we will take some questions at the end of our talk today. So please use the Q&A button at the bottom of your Zoom screen and you can submit a question that way. My name's Eugenia. And before we start our proceedings today, I would like to acknowledge and pay respect to the traditional owners of the land on which the University of Sydney is built, the Gadigal people of the Eora Nation. And as we share our knowledge, teaching, learning and research practices within this university, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. Now, I would like to introduce to you today our guest of honour. So I'm just going to see if I can pop him up on our screen. There we go, thank you. So it's my absolute privilege today to introduce you to Professor Edward Holmes. Edward is an ARC Australian Laureate Fellow and Professor with the School of Life and Environmental Sciences and the Sydney Medical School here at the University of Sydney. Now, he was elected a Fellow to the Australian Academy of Sciences in 2015 and also of the Royal Society in 2017. And that same year, Eddie won the New South Wales Premier's Prize for Science and Engineering in the Biological Sciences. He's known best of all for his work on the evolution and emergence of infectious diseases, particularly the mechanisms by which RNA viruses jump species boundaries and emerge in humans and other animals. Now, he's studied the emergence and spread of pathogens such as the influenza virus, dengue virus, HIV, hepatitis C virus, and most recently, SARS-CoV-2. So as you can probably deduce, Professor Holmes's knowledge and skills are very highly sought after at this time. So Eddie, we really appreciate you joining us today for Lunchbox Science. So now I'd like to hand over to you, Professor Edward Holmes, to talk about viral epidemics and the emergence of COVID-19. Thank you very much, Eddie.
Thank you, Eugenia. I'm just going to share my screen. Right, so thanks all for coming this lunchtime. So if you can see, there's a very slight change in my title of my talk there. And rather than using the word viral epidemics, I've replaced that with the word zoonoses. And the zoonoses, or zoonotic event, is when a pathogen jumps from an animal to a human. And today I'm going to talk about zoonoses in general, so it won't just be viruses, there'll be some other things in there as well. And I'm going to use that as a backdrop to think about the most recent zoonotic event that we're talking about, why I'm in an isolated office and on the Sydney campus, and that's COVID-19. So zoonotic events will be the kind of this jumping of species boundary, will be the kind of setting for what I'm going to talk about. Now, to start this, I'm actually going to wind the clock back a couple of thousand years to ancient Rome. And obviously the Romans did a lot of things for us. And one great thing they did was bury their dead in some very easily accessible necropolises. So these are kind of Roman burial temples you find across ancient Italy. Now I'm raising them because back in ancient times, ancient writers, so people like Hippocrates and Galen, they described a disease during the time of ancient Rome that resembled malaria. Okay, you're all familiar with malaria today, but they described it as kind of the symptoms of fevers that we associate with malaria back a few thousand years ago. And so one of the great debates in the history of disease has been, was malaria really in ancient Rome? Was it in that empire, what we now think as Italy? And did then the Romans did they remove malaria perhaps by controlling swamps? And so this is work with Hendrik Poinar from the University of McMaster in Canada. What we did was, with some archaeologists, was go to a variety of these necropolises in Italy and look at human remains, basically teeth of in these burial sites. We looked at 3. So one's called Iola Sacra, and that's the one you're seeing in the picture there. This is basically near the port of Rome. That's that one there. One called Velia, that's south of Naples, and one called Vagnaria, that's in Puglia, agricultural region in southern Italy. So we took teeth from these individuals who are buried in these necropolises, and these dates, it's not thousands of years old, but they date to about 2nd to 3rd century AD, so about 2,000 years old. I'll show you what the remains look like. I know it's your lunchtime, but it's good to see. This is an individual from Vagnari, from that site there in Puglia. And this is a male, about 35 years old. You can see the teeth, okay, very clearly. And so what you can actually do is take a tooth, and you can saw the top off, and you drill down. In that tooth, there's remains of dental pulp. And that dental pulp actually contains, amazingly, lots of microbes. So then you can sequence the genomes of all the microbes there, and you find out what they are. And we did that, and miraculously, we found that in the pulp of these remains from these 2,000-year-old Roman sites, we found the genome, the sequence of Plasmodium falciparum malaria. This is the parasite that causes the most severe form of malaria. It's actually in the teeth of the 2,000-year-old Roman remains. And then you can do an evolutionary tree to understand, to place that sequence in the context of all the known types of malaria. So this is an evolutionary tree here. So those of you that are not used to evolutionary trees, very briefly, an evolutionary tree is exactly the same as a family pedigree. Well, we all know about family trees. So here's a kind of cartoon one here. We've got some brothers and sisters, a brother and sister here, because they have parents and they have grandparents. And that evolutionary tree just gives you the pattern of descent through time of those individuals. And an evolutionary tree over here is exactly the same thing, but stretched in evolutionary time. So rather than 2 generations, we have thousands of generations. And also you can draw the trees in different orientations. So here I'm going from, vertically. You can also go horizontally from sort of left to right. If you look at that tree over here in the grey box, these are the Plasmodium falciparum strains. That's that parasite that causes the most severe malaria. And the one in red is the Roman one. So that's very clearly the Romans had very bad malaria in ancient Italy a few thousand years ago. Now, what's also interesting is these other strains here that got kind of unusual names. Some are found in humans, but these are mainly found in other animals. And what work has shown over the last 10 years or so is that Plasmodium falciparum malaria, the parasite that causes disease, has actually jumped to humans from great apes. So this is work done by Beatrice Hahn in the US. And what she's done very nicely is show that different apes, this is chimpanzees and gorillas here, have different strains of Plasmodium falciparum. And the one that's closest to humans, this is Ronald Ross, who basically worked out the malaria transmission cycle. So the one that's closest to humans comes from a gorilla, okay? So there we see a really amazing classic zoonotic event. This parasite jumped from gorillas to humans, probably in West Africa, probably because malaria has already passed on by a mosquito. So probably a mosquito bit a gorilla and then a human. And we think that happened over 10,000 years ago. And of course, then what's so striking is that although this happened in West Africa, this malaria strain has then found its way into ancient Italy, and of course globally in the preceding century. So a really beautiful example of an emerging event. And it turns out that great apes, like gorillas, have actually been responsible for a number of zoonotic events. So jumps from a pathogen from an animal to a human. I should also say that humans can also give their pathogens to animals too. So it's not just one-way traffic. So another good example of a pathogen that's jumped from an ape to a human is HIV, the cause of AIDS. And we now know, with some, again, work by Beatrice Hahn, that if you go to West Africa again, this photograph here was taken from Cameroon. It's showing you what's called bushmeat. These are animals just caught in the wild there. They're on the street to local people as food. If you go in, and there are primates in this, is obviously a monkey. If you go and look at the animals that are sold as bushmeat, they contain viruses that are basically very closely related to HIV. In particular, chimpanzees. I'm sorry, it's a very confronting image. This is a chimpanzee that was caught in a poacher's sack. these chimpanzees contain a virus that is actually the ancestor of the AIDS virus that infected humans. And what we think happened is that during the early 20th century, the logging industry expanded in West Africa. As they expanded that industry, they encountered new animals. They took those animals as food, as bushmeat, and then at some point, maybe in preparing meat, the virus has jumped from primates like chimpanzees into humans. So these zoonotic events are really critically important. And in fact, most infectious disease emergence is just the word that describes the appearance of diseases. Most of that emergence is due to pathogen host jumping. Pathogen host jumping is just a zoonoses. So they're extremely common. And you look on the web, you can find lots of pictures like this, just showing maps of the world and showing where pathogens have jumped from animals to humans. So obviously in Australia, we've had the Hendra virus that's come from bats. Obviously now in China, we've had COVID-19. I'll discuss that more shortly. And of course, this zoonotic phenomenon is now massively in our mindset and all over our media and our consciousness. So, for example, there are films. You've seen Contagion, about a kind of bat virus that spreads in humans. Not too bad, a bit exaggerated for Hollywood, but pretty accurate. There are books. A book by David Quammen called Spillover is a really good book, actually, the best I've read about zoonotic events. And David's actually writing a book on COVID-19 that will come out in a couple of years. He interviewed me the other day, so hopefully that'll be quite good. And what my research is, what my group do here in the University of Sydney, is we try and understand what viruses there are in nature, so in apes and monkeys and whatever, and how they can jump to emerge in humans. And the way I like to do this, I like to use a very simple analogy from earthquakes. So where humans and animals interact, that you can think of as like a fault line, same as two plates interacting in kind of, in earthquake science. So those interaction zones, those fault lines where humans and animals kind of share space, that's where pathogens can jump between them, okay, obviously. So what we do is we sample those places, those fault lines where animals interact. So we can sample domestic animals, livestock, companion animals, and most importantly, places where humans have encroached onto the animal world and we encounter animals. What kind of things get passed between humans and those animal species? And we sample those. And the way we sample, and this is not very technical, just very quickly, we use a technique from genomics, from genetics, called metatranscriptomics. Meta just means all, and transcriptomics describes the transcriptome. So what that means is we are sequencing the RNA in a sample. You take a sample, it could be a tissue sample or a fecal sample or blood sample, whatever, and you sequence all the RNAs. Again, DNA makes RNA, and we sequence that RNA. And then once you have the RNA, we work out which of the RNA is from the host, so it's from a human or an animal, and which is from a microbe. And the great thing about this metatranscriptomics, I mainly work on viruses, I should say, but if you have, for example, this is like a tick bite in a person here, this metatranscriptomics will give you all the bacteria, fungi, parasites, or viruses. So it gives you everything. And the real power of it is other techniques we use to discover new pathogens can only detect a tiny proportion of total pathogens that are out there. So for example, viruses, the total universal virus that I like to call the virus sphere, every virus out there, okay? Now the way many people try and work out what the virus sphere looks like is they use a thing called cell culture. You can imagine evolution like a schematic evolutionary tree here. This tree is all the possible viruses there are. The red blobs are what cell culture can find of that total virus sphere. It's a few twigs, a tiny bit of it. PCR, that's what we use at the moment to test for COVID-19 infection. That technique can get you a bit more of the virus sphere, but this metatranscriptomics, that's the kind of orange thing there, can potentially get all of it. And in doing that, what my team and others have shown, and I won't go into any detail, is that we think there are an enormous number of viruses out there. The virus sphere is absolutely enormous. And my guess is and it really is not much more than a guess, but is that there may be more than 100 million viruses out there in eukaryotes like us alone. So you can imagine this is like this grey circle here. That's the total universal viruses. The ones we found in humans are that tiny little dot. So to date, there's about 200, 250 human viruses. So a tiny fraction. Of the viruses that we've discovered so far, here I've got 4,500, it's probably now about 6,000 viruses, but still a tiny fraction. So the vast majority of the virus sphere is completely unknown, okay? And it's no surprise that some of those things jump species to cause disease. Now, I'll just give you one example, and I'll come back to this a bit later on, so I'm going to show it now, of another virus or viruses that have jumped from animals to cause disease in humans. And these are viruses that cause hemorrhagic fevers. So hemorrhagic fever, as you might expect, is a fever, and it can cause a hemorrhagic disease, so capillary leakage and bleeding. And when that happens in an internal organ, that can be quite serious. And these hemorrhagic fevers are caused by a variety of different pathogens. So Ebola would be a hemorrhagic fever. Hantavirus is called hemorrhagic fever, as the Bunya virus. I won't really mention what these are, but they're RNA viruses. And here's a very good example of a person here who's suffering from a hemorrhagic fever, this kind of capillary leakage there you can see. And this is caused by a Bunya virus that comes from ticks. I can never really ever say the name of that virus, but anyway, it's a tick-borne Bunya virus. And we think hantaviruses, in particular, and I'll come back to these later on, these we think come from rodents. So lots of mice and rats and things like that carry these things, and they excrete these viruses in their feces and urine and saliva. So if we come into contact with those secretions, we can then get infected. These things are actually very serious, and they're in North America, they're in Asia, they're in Europe, and they do quite often result in human fatalities. And so the way we go and sample these to try and look at what's out there, into basically villages and towns and capture the rodents that are close to them and see what they have. This is the work done in China, and this is taken from my phone at kind of safe distance. And then my colleague has just collected a rat that we will then sample to see what viruses they have in them. So as well as rodents, we do quite a lot of work on bats. Okay, this is obviously very important at the moment with COVID-19. And so what we've done, again in China, I have lots of colleagues, is you go to these rural parts of China. Here's a little photograph I took from my phone, a place in Zhejiang province in the southeast of China. Very interesting kind of step terracing there you can see. And then unfortunately, they take you to a cave and they say, okay, put on some PPE, you're going to go in. So you got to crawl through, that's a struggle. You got to crawl into these caves. And there's me just to prove I was there. Those interested, I did actually have goggles on, but they're kind of steamed up to take the photograph. So that's me in the cave collecting bats. And in doing that, we found a very large number of coronaviruses, of course that's why we're here today, in bats. And so here's a paper published a couple of years ago in a journal called Virology by some Chinese colleagues who described some of these bat coronaviruses. And why I put it up is because right at the start of the paper, we say the following thing, that, and I'll just read it out to you, the ongoing emergence of these COVs, coronaviruses in humans, means that coronaviruses will likely remain a key public health threat for the foreseeable future. So myself and many people were saying these coronaviruses are a problem. They could jump species, they could cause disease, we need to watch them. So, of course, COVID-19, that's why we're here. So now I'm going to talk you through a few slides about what we know at the moment about the early emergence of this virus. And unfortunately, there's still quite a lot of questions out there, but I'll just try and paint as much as I know. So the disease is called COVID-19. It's caused by a virus called SARS-CoV-2. And the first I heard about this, as did many people, was through a website called ProMed. So ProMed is a society of infectious diseases. And every day they post on Twitter, on their kind of website, lists of ongoing disease outbreaks in humans and other animals. So here's one they posted basically midnight on the 30th of December last year. And it describes an undiagnosed pneumonia in China. They talk about some of the clinical symptoms here. And they say they've got only four cases, right? The first person with unexplained pneumonia appeared in Wuhan and associated with the seafood market. So that's the first that I heard about it. And then the next day, my colleague, Peter Daszak from New York, he tweeted about this and he links the ProMed link. That's the ProMed link there, so he lists that there. And I retweeted this because it was obviously an issue. And Peter says, look, it has similarities to SARS. This is right at the start of January. then what happened? I actually have quite a lot of colleagues in different parts of China, particularly in Shanghai, Professor Yongzhen Zhang, Professor Zhang, and we had also a variety of projects in Wuhan. And I've been to Wuhan a few times. In fact, this is a picture here of Wuhan Central Hospital in 2016. And you can just see, if you look at my name there, so they're saying, welcome to me to the hospital. Actually, I was giving a talk on that at that time. So Professor Zhang and about 2 or 3 other teams in China were scrambling early on in late December, early January to sequence the genome of this new pathogen. So Professor Zhang sequenced it in early January, along with some other teams, and I then posted it on a website called Virological, and I announced it on Twitter on January the 10th or 11th, depending on your time zone. So that was the first recorded release of the genome sequence of the virus, and that's where things really started to get going. Now, as soon as we had the genome sequence, and people as well, it became very clear that this new coronavirus, we now call it SARS-CoV-2, it used to be called 2019 nCoV. That coronavirus was actually very closely related to the first SARS coronavirus. That emerged in China in 2002, 2003, and spread to a few countries. I'm sure you remember. So this is an evolutionary tree again. This is SARS-CoV-2, that's the new one. Up here, that's the first SARS coronavirus. And they're about 20% divergent, which doesn't sound that much, but for a virus, that's actually quite a lot. So that's SARS-1, that's SARS-2. What's also interesting is that these are beta coronaviruses. They're different genera of coronaviruses. These are all the genus beta coronavirus. So MERS coronavirus that emerged in the Arabian Peninsula in camels in 2015, that's that one. And there are a couple of other human ones. So we knew very early on it was a bit like SARS. We also knew that it was close to a variety of bat coronaviruses. And I've already mentioned that bats carry lots of coronaviruses. And in this tree here, the sequence is in black, the names in black, they're all from bats. You can see there's lots of these bat coronaviruses, and both SARS-1 and SARS-2 kind of agree with them. Now, as we then got the sequence, we could also look at some more details of the sequence. This bit's a little bit technical, I apologize, but I just thought I'd very quickly go through it. So this is the genome of the virus, and it's about 30,000 bases or nucleotides long, so very short. This gene is this long bit here. This is basically the polymerase, the replication enzyme. This here is called the spike protein. I'm sure you've heard of the spike protein. That's that gene there. If you blow that gene up, that's the whole gene spike blown up. It has 2 very interesting things about it. This bit here is called the receptor binding domain, and that, as its name implies, that's a bit of the virus that binds on to the host cell receptor. So viruses all have to enter host cells in a kind of lock and key mechanism. So that's the bit of sequence that dictates how the virus does that. Also here, if you can see, it says S1 subunit, S sub 2 subunit. So the spike protein is in 2 kind of bits, and at that junction, that's the junction region there. In the SARS-CoV-2, it has an insertion at that junction region, and it's inserted 4 amino acids that have a basic amino acid chemical structure. So it's proteins, arginines, and other ones. Okay, and that's called that produces what's called a furin cleavage site. So furin is a protease that can kind of cleave that bit of sequence there. And that insertion is very unusual. It's led to lots of kind of speculation about how that ended up in that virus. It is actually in some other coronaviruses, but not in the ones most closely related to SARS-CoV-2. To cut a long story short, there's been a very nice work by a guy called Bill Gallagher from New Orleans, and he has shown that what's most likely happened is that furin cleavage site insertion is results from 2 bat coronaviruses that have entered cells and there's been some slippage between them. It's called what's called copy choice replication. So the sequence there is basically the same between the SARS-CoV-2 and this bat virus, and that's produced this furin cleavage site insertion. That's the most likely theory at the moment. Now, the next question is, where's the virus come from? So another thing you've heard about a lot already, and I've mentioned it once already, is this very famous seafood or fish market in Wuhan. And I got very interested in this because I've actually been there. Okay, I went there about five or 6 years ago. I took some photographs of the market, and it's the name fish market is a bit apocryphal because there's lots of different animals in there. People say it's a crowd. It's like a set of alleyways with lots of stalls in. It wasn't actually that busy when I went there. This is taken from my phone again. But there are some amazing things that you can find on sale. These are some rodents. This is actually a raccoon dog. These are cages on the floor in the market. So the big question is, what role did the market play? Was it the cause? So did the virus actually jump from an animal like these rodents, these raccoon dogs, to humans in that market? Or is it just a consequence? Did it just happen to detect the first cases there and actually emerge somewhere else? And at the moment, we really don't know. Now, we know that some of the first patients were associated with the market, but others are not. I'm interested in it because this is exactly where you predict a host jumping zoonotic event to take place. In fact, I was taken there in 2014 by the local people because they say, look at this place, it's where we think something could happen. And lo and behold, it's where we detected SARS-CoV-2. We also know that environmental samples, so surface swabs, are positive for the virus in the market. But the latest statements at the moment suggest that none of the animals in there they've looked at had the virus. So at the moment, we really don't know whether it played any role at all. And it's a complete coincidence the virus turned up there. And we just need more data to resolve that. We also don't know exactly what the animal reservoir is. Now, this is quite a busy slide, I apologize, but it's quite a lot of information on here. So I've already told you that coronaviruses are very, very common in bats. And just yesterday, over the weekend, Peter Daszak's group posted a paper on bioRxiv describing lots of new bat coronaviruses. This is basic evolutionary tree drawn in a kind of a different kind of way. All the way to see is the blue colors, those viruses, this is all the coronaviruses related to SARS-CoV-2, all those coronaviruses are from bats, particularly these horseshoe bats of the genus Rhinolopus. So these bats here, these are horseshoe bats, and they have lots and lots and lots of coronaviruses. You look at SARS-CoV-2, see there's blue dots very close to it. That's 2 coronaviruses from these horseshoe bats, very close to SARS-2. Also, amazingly, pangolins, these very endangered, highly trafficked, vulnerable mammals, also have a closely related virus to SARS-CoV-2. They're the ones in green. And in that receptor binding domain I mentioned earlier, the pangolin sequence is almost identical to the human sequence. So we know that pangolins and bats, horseshoe bats, have viruses very close to SARS-CoV-2. But although I said they're very close, in evolutionary terms, they're still actually quite a bit different. So we think there's still at least 30 years of evolutionary separation between SARS-CoV-2 and the closest bat relatives. That little branch there, although it looks very short, it's at least 30 years or more of evolution. So there is a gap in our understanding of where this virus comes from. Now, people are, I guess, a bit frustrated by this, but just need to remember this. We now know that bats are the cause of SARS-CoV-1, the first SARS coronavirus. It took 2 years for us to show that bats were the cause of that virus. And it took 10 years or so to show that chimps were the source of HIV. So we've only been doing this for like 6 months yet, so we need to be a bit patient. I'm sure we'll find out what the reservoir is. We're not quite there yet, but hopefully we'll get there in the near future. Now, as the virus has spread from Wuhan in late December, it started to diversify genetically. And so you can do an evolutionary tree of SARS-CoV-2 that spread globally. So here's a picture of the tree. Sorry, it's not very exciting to look at, but it is actually quite interesting. And if you look, you can see there are two sort of major clusters of the ongoing evolution of SARS-CoV-2, the red and the blue, that myself, Andrew Rambo and Oliver Pybus have very cleverly called lineages A and B. And we think they separated, as most people, we think they separated round about November 2019. So that's our best guess, around about November last year, when we think this virus first emerged. There's an error bar there, but that's what most estimates put around about November last year. And you'll also hear lots of discussion about different strains maybe differing in how virulent they are, if they can affect people better than others. And at the moment, there's no really convincing evidence for that. There's some suggestions, people are trying to work it out better in the lab at the moment, but We're not entirely sure. So at the moment, all the viruses look pretty similar in their basic kind of phenotype. A couple more things that are quite interesting. So these lineages A and B, they differ by just 2 mutations. Most of those wet market ones, they are found in this lineage B, that big one there. Of the B ones, the most interesting one is a lineage we call B1. That's that big cluster there. And that one has really spread globally. And that one we think appeared in Italy and then spread from Italy to many other countries. And it contains this very famous mutation called G614D in the spike protein that some people have suggested has made the virus more infectious. Again, we just don't know that's true or not. The jury's out on that one, but watch this space. In Australia, we have a lot of a virus called B4, which I think is up there somewhere, not quite sure which one. And this virus in Australia, we think came from Iran. So China to Iran, Iran into Australia. So coronaviruses, I told you, have lots of bats carry coronaviruses, and they are in many other animal species. So why is it that coronaviruses seem to be able to be very commonly zoonotic agents? And here's a slide I made back in 2018, so way before the pandemic started. I want to show here is, look at this tree. This is a tree of the hantavirus I mentioned a bit earlier. And this is a tree of the virus, and this is the tree of the hosts that they're from, the different mammals and like rodents and bats and things. Now, you see the trees are kind of almost like mirror images of each other. So the virus tree looks like the host tree. And that tells us that, in fact, there's not many jumping events going. So these viruses don't tend to jump between hosts. You can see the ones that are kind of the diagonal lines. That's a host jumping event. Most of it is pretty similar. There's not much jumping around. If we then take a tree of coronavirus, it's the tree I made in 2018. So there's no SARS-CoV-2, there's SARS-CoV-1, there's MERS. You can see there are lots of other animal symbols. So there's deer and there's pigs, rodents and bats and whatever. And if you then compare the host tree and the virus tree, look at that. Amazing, right? There's lots and lots and lots of jumping going on. So back then, in 2018, I wrote in this slide saying, look, these things really jump host frequently. Is therefore emergence more likely? So why coronaviruses? Why do coronaviruses seem to jump so commonly? And here's a nice picture of coronavirus for you. So it turns out that SARS-CoV-2 is the 7th human coronavirus documented in humans to date. Of those 7, 4 cause common colds. Okay, they got strange names: 229E, HKU1, NL63, OC43. And you and I all have a few of these every year. They're very common. They go around everywhere, right? 3 of the 4 cause serious disease. Okay, SARS, first one, MERS, in camels, and SARS-CoV-2. Amazingly, 5 of these coronaviruses, 5 of the 7, have emerged in the last 20 years. So SARS, MERS, HKU1, NL63, and SARS-CoV-2. And bats are involved in also the emergence of 5 coronaviruses. The reservoir host of 5 coronaviruses: SARS, MERS, SARS-CoV-2, NL63, and 229E. So why? Now, we don't really know why coronaviruses have this amazing ability to jump hosts and spread. It could be that they're mainly passed on by respiratory particles, and sneezing, whatever, and that may allow them to get into new hosts easier. It may also be because they use receptors, those lock and key things on the cells that are quite conserved in evolution, so different viruses can recognize them. We don't know, sure, that's kind of clearly a big thing to investigate. But I think it's very, very likely that SARS-CoV-2 will become endemic in humans. So though we're having an epidemic, a pandemic now, this is not going to go away. We'll probably have this forever. I can't see we'll ever eliminate this virus. It'll be around as long as humans will be, almost certainly. And so of these 7 coronaviruses, 4 are currently pandemic: HKU1, NL63, OC43, and 229E. And I suspect SARS-CoV-2 will be one of those. So how can we stop this ever happening again? And again, here's a little quote I made from a talk in 2015. I'm saying these things will jump, pathogens will jump, we will get more pandemics in the future. So how can we stop it? So I think there are a few things we can try and do better. So one, and I've written these in kind of order of hardness. So one, we need to try and limit our exposure to wildlife. So I've already mentioned this wet market. Now, again, it's unclear whether the wet market was involved at all. We just don't know. The wildlife trade, that could also be important. Zoning. So one of the big problems is that humans build their habitats very close to animal populations, and we expose ourselves. A very simple way, it turns out, that you can stop Nipah virus emerging is to plant more fruit trees away from villages, and the bats go to those rather than go to villages. So we need somehow to reduce our exposure. It should be a simple thing to do, although obviously people's livelihoods depend on wildlife trade and wet markets, so that needs to be worked out as well. But first, we need to limit our exposure. Second, we need to establish a much better surveillance mechanism using genomics, I mentioned those, and serology, and also social media to really work to look at what's happening at these fault lines, at this human interface. So, for example, ProMed, this is a map from ProMed. They have a little heat map, little alert map with flags in the territories that are actually where you're seeing events going on. This is a photograph I took from a village in China in 2013. This chap here is a local kind of CDC officer. He's written on the board there in Chinese about an H7N9 flu emerging in chickens. He's telling people to kind of watch out for that. So this is very local level surveillance, but we can make a much better system infrastructure to do that. And thirdly, and hardest of all, we need to be better prepared. We need to try and develop and stockpile broad cross-reacting antivirals, so drugs and vaccine. Very easy to say, very hard to do. So at the moment, we're trying to make a universal flu vaccine, proving a challenge, but one day we will get there. But we need to have in the freezer cross-protective vaccines against all the viruses, like coronaviruses, we think could jump in humans. And before you think this is kind of an alien thing to Australia, we do get these zoonotic events here as well. So a few years ago, we started working on a disease called Tularaemia. It's a bacterial infection caused by a bacterium called Francisella tularensis. There's a die-off of ringtail possums over here in St. Ives in 2003. We did some genomic work, and we found out that they carried the bacterium that causes Tularaemia. And Tularaemia can cause disease in humans. It's got a nasty disease. And just 10 days or so ago, you may have heard that a woman from northern Sydney was bitten by, I don't think she was actually attacked by a possum. I think she picked up a diseased possum. Possum was sick, it bit her, and she got Tularaemia. So it's there, it's in wildlife. We need to reduce our exposure, not pick up these things, and then not get these diseases, but they're there. So I think that's kind of mainly what I want to say. So obviously lots of people are involved in this work. I haven't really got time to mention them all. I just want to mention again, Professor Zhang from Shanghai, who's really done some amazing work on this. So this is a photograph. When I went to Shanghai Wuhan Central Hospital, they took me, that's me there in my head, they took me to, that's Dr. Zhang, they took me to the respiratory diseases, infectious diseases ward. And there it is. That's the ward in 2016. You can only imagine what that looked like in January and February of this year. Really quite extraordinary. So these people really are what they did there was really quite heroic. I think I'm going to stop there actually. So questions? Oops.
Thank you so much, Eddie. That is absolutely fantastic. Thank you for joining us for Lunchbox Science today. And I will just mention as well, if anyone does have any questions, please use the Q&A button at the bottom of your Zoom screen. And we'll try and get to a few questions now, if that's okay with you, Eddie. We've got a few coming up.
I'll just read them out, okay?
We've got a few listed here for you. So first one surrounds the idea of viruses and seasonality, actually. So we tend to talk about the winter months being a time where we have lots of colds and flus. And there's certainly been some speculation around, in the Northern Hemisphere, at least, when we go into the warmer spring and summer months, that maybe this virus will sort of peter out. Is that the case? Will we have the severity or the spread of this virus sort of decreasing with warmer, sunny weather?
So the general rule is that although many respiratory diseases like coronaviruses and flu are seasonal, when you have no immunity in the population, seasonality does not normally occur. So seasonality develops develops as people start to get immune in the population. So one of the really bad things, there are two really bad things about SARS-CoV-2, COVID-19. One is it's obviously very, very nasty, and the fatality rate is way higher than flu. Also, no one is immune. Everyone is susceptible, okay? So when everyone's susceptible, the virus can very easily find susceptible hosts and infect them and keep passing it on. And in those circumstances, seasonality is not so important. As people get immune to the virus, my prediction is seasonality will develop. And it may take a year or two to kick in, but I suspect it will be probably a winter disease in a while, but maybe not this year.
Okay, thank you. There's a question here actually around MERS. So given CoV-2 causes this really severe disease similar to MERS, has MERS disappeared? Is it still out there? And how do you think CoV-2 will remain endemic? And do we know what's happened to MERS?
So MERS is still around. So MERS is actually much more serious than SARS-CoV-2. So the fatality rate for MERS is probably over 30% of infections, case fatality rate. So it's really quite extraordinary. Whereas for COVID-19, we're talking between 1 and 1.5%, depending on your age group and what country you're in. But that's the kind of, so that's which is very, very high, I should say, but not as high as MERS. MERS is very common in the Arabian Peninsula. It's very common in camels, and camels can pass it on to humans. Now, the good thing about MERS, although it's still there and it causes the occasional disease in that part of the world, it's never really evolved efficient human-human transmission. It's really only ever spillover infection. And the problem with SARS-CoV-2 is it is a very, very, very good human pathogen, and that's the problem. So if you combine that with a pretty high fatality rate, it's a serious thing. But MERS is still there, and I think SARS-CoV-2, it's so infectious, and it's all over the world. Until we eradicate it everywhere, it's always likely to come back. And I cannot see, even though Australia's done a phenomenal job. I mean, we're very blessed to live here, right? We're a phenomenal job, as of New Zealand. I can't see how globally it could ever be eliminated. It's just too infectious.
Yeah. Okay, so just following on from that a little bit, I guess, we have another question here. Are there any sort of platforms that you could suggest for the surveillance of infectious diseases? What can we use?
Well, so there's lots of, I mean, each country will have its own surveillance system, and they will vary in how good they are. ProMed is a great thing. ProMed is a kind of volunteer ad hoc thing that people use. And of course, overarching that, we have WHO. And one of the problems, I don't want to get into politics at all, if I can avoid it, but one of the problems about this disease is it has become extremely politicized very, very quickly. And so somehow we need to break down those barriers and build something that integrates all the different ways we can do surveillance into some kind of body or institution that is allowed to work in every country equally, and it has, you know, complete flexibility. You can bet your bottom dollar, once this is over, or once we've declared, you know, countries have controlled it, there will be things put in place. There's no doubt about it. We cannot let this happen again. And so I think we'll build a completely new surveillance system in the next 5 years or so. What that will be, I don't know. And quite who will run it is unclear as well. But we obviously have to do a lot better than we've been doing.
Yeah, it sort of reminds me a little bit of, you know, the steps that came in, all the surveillance that came in place for tsunamis. So, you know, we can develop the technology.
Yeah, no, I think we can.
Another question for you from Stephen. Is there any evidence of cross-reactivity between these coronaviruses and therefore any cross-protection from previous coronavirus infection? Interesting question.
Yeah. So as I said, there are lots of coronaviruses out there, and there's a couple of the ones that the common cold ones that are quite closely related to SARS-CoV-2, the beta coronaviruses. A lot of people are trying to work that out at the moment. as far as I know, there's no good evidence of cross-protection. I've not seen any compelling evidence of that at the moment, but that's certainly something that people are actively trying to work on. But at the moment, it seems to be that pretty much everyone is susceptible. And obviously the older part of our population suffer the worst disease. So I don't think at the moment there's any really strong evidence of cross-protection, but again, it's a massive area of interest at the moment.
All right, another one here. Something about adaptability and evolution, I suppose. Is the virus able to adapt, resulting in different strains? And can new strains reinfect people that have already had the first or original strain, if you like?
So, yeah, so the virus is evolving at the same rate as any other RNA virus. It's ticking along. It's evolving slower than flu. Flu evolves about 4 or 5 times faster than SARS-CoV-2. But SARS-CoV-2 is evolving about the same rate as most other RNA viruses. As it evolved and spread, it could, in theory, adapt to our population. I mean, it's pretty well adapted as it is, actually, I should say, but it could evolve to become more infectious. It could evolve to become nastier. It could evolve to become benign, right? We really don't know at the moment. Any of those things is possible. As time goes on, at the moment, most of our population is still pretty susceptible. We look at Europe, the sero surveys are talking no more than 10% really in different countries have been infected, been exposed to the virus. So the vast majority of people have not been exposed as yet. But as the people who've been infected, it rises and immunity starts to appear, I suspect the virus will start to evolve antigenically. And there'll be, as immunity will push the virus to change and change its coat proteins to escape, as most viruses do. So I expect that to happen, not this year, maybe next year when many more people are infected, or maybe we have a vaccine. Hopefully that will come in the next 12 months. And the vaccine will also push the virus to kind of adapt away from it. So I suspect the virus will certainly evolve in that way. But that's nothing to worry about. That's a kind of standard virus evolutionary thing that we do.
I know there's so much science, so little lunchtime. So we might just take one more question, Eddie. We have one from David. Given how many coronaviruses there are, why aren't there more emergence events?
I think there have been 5. So let's think about this. There have been 5 in the last 20 years that have taken off and spread between people. There's also all the others that just cause spillover events and never get going. So people have looked in southern China, tested people who live really near some of these big bat colonies. And I think about 3% of those people have antibodies to bat coronaviruses, right? So I think they're spilling over all the time, but they usually don't get going either because the virus isn't quite adapted properly to humans, or that one person's infected and then they don't, there's some degree of social distancing by chance going ahead and they don't infect anyone else and it dies out. So I think these events are, this traffic, these fault lines are always there. And all we're detecting are the ones that have been successful, really successful. The ones that have been a bit successful, we may never know, right? We may never see them because they're hidden. In a city the size of Wuhan, if you had someone with a mild cold, would you ever know? Would you ever test it for being a coronavirus? No, you wouldn't. Okay, and only because they had a cluster of pneumonia cases, they actually did any real work on it. So I think it's a continual process.
Excellent. Thank you so much, Eddie. Thank you for the talk and for answering those questions. We do have so many questions, and I do apologise that we're not able to get to more of them. But yeah, there are some amazing questions in there. So we'll be sure to show Eddie what everyone's talking about. All good, all good. Now, I will let you know that a recording of Professor Holmes's talk will be available on our website, so sydney.edu.au/science. And don't miss the next Lunchbox Science. In 2 weeks' time, we have Professor Zdenka Kuncic, and she's going to be talking on Wednesday, the 17th of June, same time, 12 p.m. And she's going to be asking the question, is intelligence physical? So bit of intrigue for you for next time. And registrations are open for that event right now. So you can once again go to sydney.edu.au/science. And thank you so much, Eddie. And thanks everyone for joining us. Catch you next time.